1863, the National Academy of Sciences in the United States was chartered. President Lincoln approved the act in Congress, and the act stipulated that the Academy would, whenever called upon by any department of the government, investigate, examine, experiment, or report upon any subject of science or art. This was 1863. Its first president was Alexander Dallas Page. But today is about redox and oxidant stress. So we remember Elmer McCollum, born this week in 1873. He was an American biochemist who originated the letter system for naming vitamins. In fact, he discovered vitamin A, vitamin B. He worked with others to describe vitamin D. And he performed extensive work on nutrition and growth and the role that these vitamins had on the growth of animals. We also remember Hertgard Herzberg. Hertgard Herzberg died this day in 1999 at age 94. He was a German-Canadian physicist who was awarded the 1971 Nobel Prize for Chemistry for his contributions to the knowledge of electronic structure and geometry of molecules, but his main focus was in free radicals. Okay? But perhaps the person that brought this whole idea of antioxidant and oxidants in disease and health, who become a widespread issue, was Linus Pauling, who lived from 1901 to 1994. He was the only person to actually win 2 unshared Nobel Prizes, one for chemistry in 1954 and one for peace in 1962. I bet you didn't remember that. And because of that reputation, whatever Linus Pauling said, people just believed, okay? And because of him, there was this widespread misbelief that high doses of vitamin C are effective against colds and other illnesses like cancer. And in fact, a number of rigorous trials of antioxidant supplements in people have not found that high doses prevent disease. The Women's Health Study studied 40,000 women, showed that vitamin E does not reduce heart attack, does not reduce stroke, cancer, age-related macular degeneration, cataracts, or overall death. The Women's Antioxidant Cardiovascular Study studied 8,000 women. Vitamin C, beta-carotene failed to affect heart attacks, strokes, death by cardiovascular disease, or the likelihood of developing cancer. The Physicians' Health Study studied 14,000 male physicians over age 50, found no effect of vitamin E or C in reducing cardiovascular events, cancer, or cataracts. And Selenium and Vitamin E Cancer Prevention Trial investigated 35,000 men and showed that vitamin E did not prevent prostate cancer, and it could perhaps enhance it. So there's strong evidence in vitro and in animal models, and even in some data in humans, that suggests that oxidant stress has important roles in health and disease. And yet we can't figure out how to intervene to take advantage of this knowledge to help people. And I must believe that this is because we are too naive, and that the complexity of this issue is just so tremendous that we think like a little pill like N-acetylcysteine bought at GNC stores for $150 to $100, that you can survive all these conditions because you switch that antioxidant-oxidant ratio. And so, because patients come to us all the time saying, we're taking all of this, and which one would you recommend, Dr. Roman? I thought it was time to bring somebody to the stage to get us back to medical school and chemistry in your days of college and tell you about oxidant stress and redox stress, okay? And for that, I bring to the stage Dr. Walter H. Watson. Dr. Watson obtained his undergraduate degree in chemistry at Emory University. He obtained his Ph.D. in toxicology at the University of Kentucky in Lexington. He went back for his postdoctoral fellowship to work with Dean P. Jones. Many of you have heard that word as someone who's interested in redox stress as well as metabolomics at Emory University. Then he was assistant professor at Hopkins for a while, and fortunately he came to the University of Louisville in 2010, where he is assistant professor of medicine and also in the Department of Pharmacology, Toxicology. He is on the editorial board of Journal of Nutritional Biochemistry. He has served as course director of a number of toxicology courses here in town. And his work in the area of oxidant stress is collaborative, and he's working with other groups in lung diseases as well as with the GI group. He's part of the P50 and COBRA grants, among others, at this organization. And he's going to take us back to that first year of medical school to tell you why we are not smart enough to take advantage of our knowledge of oxidant stress to help people. With you, I leave you, Walter Watson.
Well, thank you, Dr. Roman, for setting the crowd up so nicely. I think you might have lost them when you mentioned chemistry, but we'll do our best. There is a little chemistry. I apologize. I noticed that in recent grand rounds, I haven't seen a lot of Ph.D. investigators talking, and so I think this is the first time in a while, and hopefully it won't be the last time. But I think Dr. Roman really did a nice job of setting up what the problem is, that we know oxidant stress is involved in a lot of things, and we're not, as he said very accurately, smart enough yet to take full advantage of it. And so what I've been working on for the last 15 or so years is to try to learn more about these systems so that we can hopefully develop the knowledge to the point where we can take advantage of it. And we're starting to make some headway toward that, and that's why I'm here to talk to you so you can start thinking about some of these concepts as you go into the clinic. So as I understand, the tradition at Grand Rounds is to open with a case, and since I don't have any cases, I asked Dr. Roman to provide me one, and he came up with one that's very interesting. He had a 66-year-old Hispanic male, stopped smoking 4 years ago, came in with chronic fatigue that had been getting worse over the last couple of years, same with a dry cough. And on physical exam, saw that he was on oxygen supplementation. He had bibasilar pulmonary crackles, clubbing in the fingers, and the pulmonary function test showed there was some restriction. And CT scans, I'm told, show classical signs of idiopathic pulmonary fibrosis, and the pictures are so great, I wanted to show them to you. So there was a trial of patients with idiopathic pulmonary fibrosis, the Panther trial that Dr. Roman and others were involved in. And in that trial, they were giving N-acetylcysteine as well as some other drugs to try to treat these patients and slow or prevent the progression of fibrosis. And in a subset of those patients, they actually took some blood and measured the plasma redox potential. And these are the patients that were collected. Power analysis wasn't enough to do anything with, but you can see that for the large part, most of these patients are 10 to 20 millivolts more oxidized than healthy controls, which run around minus 80 millivolts. We're going to now sort of develop and show you what that means and how we know what that is and how you can potentially apply it. But I need to back up a little and go back to med school and so that we can start to answer this question of can we use these redox states to inform our decisions about patient care. So I'm going to start with some background on what we know about oxidative stress, probably a concept that's familiar to most of you. Redox regulation maybe not so much, so we'll make that transition for you. I'll go through some of the stuff that we've done in the laboratory, looking at how these redox regulating systems are involved, and I'll give you 3 specific examples of things that we've been doing in the lab. And then I'm going to switch to looking at extracellular redox regulation, is really what we're looking at in this previous slide where we're talking about plasma redox potentials. And I'll tell you some of the laboratory studies we're doing in that regard and some of the clinical studies that we've now have ongoing with some of the people here in the room. And hopefully close with some thoughts about how to move forward into the future. All right, so our current working definition of oxidative stress came to us from Helmut Cease 30 years ago or more, where he defined oxidative stress as a shift in the balance between pro-oxidants and antioxidants in favor of the pro-oxidants. And so that can occur from having either higher levels of oxidants or lower levels of antioxidants. And you can see evidence of this in lots of chronic diseases. So simply putting in more oxidant or more antioxidants doesn't tend to fix the problem. So what is the problem? So let's talk at the reactive oxygen species. now for a little bit, these are really the drivers of the oxidative stress. It's a catch-all term that you see a lot. I want to remind you what it means. And here it basically results from the successive one electron reduction of molecular oxygen. Remember, a reduction is a gain of electrons. And so if we add one electron to oxygen, we get the superoxide anion radical. One more electron gives you a hydrogen peroxide, which is not a radical, but it's still reactive enough to react to some of the things that we're talking about in subsequent slides in the talk. Another electron gives you the most reactive of all the species on the list here, the hydroxyl radical. It's so reactive that it reacts with the first molecule it comes in contact with. And then finally, one more electron, and we have water. Now, fortunately, we have enzyme systems designed to take care of some of these things to protect us from it. The superoxide dismutases will metabolize superoxide to hydrogen peroxide and oxygen. But the hydrogen peroxide, as I mentioned, is also reactive, so we have to get rid of that. We have a number of enzyme systems that are designed to catalyze this 2-electron reduction of hydrogen peroxide, the catalase that you've probably heard about, as well as the glutathione peroxidases and the thyrodoxine peroxidases called the peroxyredoxes. We don't have any enzymes that catalyze the formation or the elimination, excuse me, of the hydroxyl radical, because you don't want to form any of that. And an enzyme to get rid of it would be unnecessary, since all you have to do is throw something at it and it's gone. But you can form the hydroxyl radical non-enzymatically, especially in the presence of free transition metals like iron. And so that's one of the reasons why the body is designed to keep free, unbound iron levels incredibly low. If you have a situation where iron is released, it can catalyze this type of reaction, and through Fenton chemistry, give you the hydroxyl radical. So where did all these reactive oxygen species come from? They come from a lot of different places inside the cell. They come from NADPH oxidases in cell membranes. They can be activated by growth factor receptors or cytokine receptors binding to their ligands. They can come from electrons leaking from the electron transport chain in the mitochondria, in the peroxisomes, and in several of the phase 2 drug metabolizing enzymes can also leak off electrons to oxygen. So I wanted to talk a little bit about the NADPH oxidases. Those were sort of the first enzyme systems that we learned about that are designed to generate high levels of oxidants. And they were discovered in phagocytes as part of the respiratory burst that is upregulated to produce antimicrobial agents to kill invading pathogens that get engulfed. And so this is an enzyme that's designed to produce reactive oxygen species for a purpose in killing bacterial infections. But it turns out there's a whole family of NADPH oxidases. They're not just on phagocytes. They're in all kinds of tissues, lots of different cell types, and even different subcellular localization. It starts to suggest to us that these enzymes may have roles in addition to simply immunity. And of course, I mentioned the electron transport chain. This is the system that's designed to capture this great amount of energy that's released when you transfer electrons from NADH to reduce oxygen to water. And if that happened in one big swoop, we'd have a flash of light and heat. But instead, the electron transport chain funnels those electrons through successive steps, pumps protons to make the gradient to make ATP. And because it's moving electrons around, there's a chance for leaks to occur. Here's an example of this being greatly accelerated by a drug that's known as a redox cycle, Adriamycin, which causes a cardiac toxicity. And it can pick up electrons from complex 1 of the mitochondria and transfer those to oxygen, which restores Adriamycin and allows it to go through another round. So you can get a great amplification of this type of system if you throw in a drug, and this is responsible for the toxicity that you see. But we have a lot of defenses against these things. Dr. Roman already mentioned some of the lettered vitamins. Vitamin E, vitamin C that Dr. Pauling was a big fan of, the carotenoids. We have glutathione, thyrodoxine, and lipoic acid. And I've got thyrodoxine as non-enzymatic. There's a debate over whether it is an enzyme, but is it a protein, but is it an enzyme? And then we also have these enzymatic defenses, the catalase, the SODs, and the peroxidases that I talked about. And I put a star by some of these. These are the thiol-dependent enzyme systems and antioxidants. And those are the ones that we're really going to be focused on as we move forward. So what happens to the ROS that escape the defenses? Well, we know that we can create a lot of damage just by hitting macromolecules and destroying them, basically. They need to be turned over. The lipid peroxidation in particular is bad because through these free radical chain reactions, you can amplify the signal and produce additional toxic mediators like reactive aldehydes. But you can also get reversible modifications, and that particularly occurs on amino acids that contain sulfur. That's the methionine and cysteine. We don't know much about functional consequences of methionine oxidation and reduction, but we are starting to learn quite a bit about the consequences of cysteine oxidation and reduction, and that's what I'm going to spend most of the rest of the talk talking about. So far, we've been thinking of ROS as bad. We have defenses against them. They are attacking the microbes. They're reactive. They react with anything. But when you start seeing these things like the reversible actions, reversible activities, enzyme systems that reverse these protein modifications, it starts to suggest to us that there may be a physiological role for reactive oxygen species in activating and inactivating certain enzymes or pathways. And that's what we're starting to see, that certainly seems to be the case, and it seems that there's key signaling pathways and regulatory molecules that are sensitive to changes in the redox state of key cysteines. And this was really first, sorry, I got ahead of myself. It's starting to emerge as a new working definition of oxidative stress. I've already mentioned our current definition that it's an imbalance between pro-oxidants and antioxidants. And, as Dr. Roman mentioned, we found a lot of diseases where you can see evidence for that type of situation, certainly in cancer and atherosclerosis, diabetes, neurodegeneration. But this definition really did lead us to try putting in antioxidants to correct. And as we heard, that doesn't really always work. Turns out it may work if the problem is you have an antioxidant depletion that needs to be restored. But it doesn't seem to be very effective in lowering reactive oxygen species levels. And it's probably the combination of a lot of things. I think you'll learn that there's more than one pathway and one type of perturbance that you can hit. But the problem may be that an antioxidant usually works only one time. It'll pick up that extra electron, and then it's done. And so enzyme systems are really going to be the key, I think, moving forward to where you can cycle and regenerate these antioxidants so they can work more than one time. And it really becomes a question of getting the drug, if you will, delivered to the right site at the right concentrations. And so a new way to think about it came out about 10 years ago when my old mentor, Dean Jones, proposed that maybe oxidative stress is really a hijacking of these normal redox regulatory processes, that you knock those out of whack. And sure, the irreversible damage to macromolecules is going to cause problems, but some of the more predictable physiological responses that we see may be mediated by these pathways that are there and designed to respond to these things in the first place. So we really started thinking about oxidants as signaling molecules in 1995 when Thornton Finkel's group showed that in vascular smooth muscle cells, stimulating with a growth factor produced this burst of reactive oxygen species that we're measuring by DCF fluorescence. And the time scale that you'll see is very short. In 30 minutes, the whole thing's pretty much gone. But if they were able to block this burst, either by putting in catalase or N-acetylcysteine, they could block the oxidant burst, and they could block this proliferative response here measured by tritiated thymidine incorporation. So that got everybody started thinking about reactive oxygen species as signaling molecules. So how would that work? So here's our scheme of hydrogen peroxide. It's being reduced to water by enzymes that catalyze the 2-electron reduction. But there are lots of other cysteine-containing proteins that can also be oxidized by hydrogen peroxide. Remember, anytime something is reduced, something else has to be oxidized because electrons are moving from one to the other. So we've got these two regulatory systems, glutathione and thyrodoxin. I've already mentioned the peroxidase systems over here that are dependent on one or the other. We also have on the other side, we have glutathione-dependent and thyrodoxin-dependent systems that restore these other oxidized protein cysteines. And in the case of most proteins, that system is going to be thyrodoxin-dependent. So what are glutathione and thyrodoxin systems? They're similar. They have complementary overlapping roles, but they're different. They both depend on NADPH for their source of reducing equivalents. Glutathione is present at much higher concentrations in the cell, so it has a larger capacity. It can also react with the aldehydes that might be formed. The thyrodoxin system is at much lower concentrations, but it has a dithiol active site, which makes it more effective in reducing oxidized proteins, and I'll show you a picture of that in a minute. So here's what glutathione is. It's a tripeptide, a glutamate, cysteine, and glycine, and its abbreviation, GSH, reflects that the business end is the side chain of that cysteine, the thiol group. And this is just showing you that if two glutathiones come together to form a disulfide, then that's the 2-electron oxidation. That's what we call GSSG. And again, it's to highlight the fact that it's a disulfide that defines what this molecule is. This is a ribbon drawing of the crystal structure of human thyrodoxin 1, where I've indicated the positions of the 5 cysteines. And this is the active site. All the thyrodoxin family members share this same structural motif, where you have a cysteine right out at the surface of the protein that can attack, and then what they call the resolving cysteine that comes and completes the thiol disulfide exchange. So if you wanted to reduce a disulfide with glutathione, you have to have 2 molecules come in successively. But with thyrodoxin, both cysteines are there. It can really do the job much better and much faster. And this is sort of a scheme showing this thiol disulfide exchange. When the protein gets oxidized, shown over here, let's say it's off in the reduced state and on in the oxidized state. It could easily be the other way around. It then has to be reduced. And so the disulfide is actually exchanged from the target to the thyrodoxin. And then that has to be reduced, and we use another protein called thyrodoxin reductase, which actually moves the electrons from NADPH. So together, the thyrodoxin reductase, and the NADPH are called the thyrodoxin system. And because it's reducing so many different substrates, it's involved in a lot of different pathways inside the cell. And I've shown here just some of the general pathways, and each arrow, say, going to gene expression. I've given an example of a protein that's reduced by thyrodoxin. Here, NF-kappa D, and we're going to talk a little bit about that later. It's involved in proliferation, apoptosis, DNA and protein repair, antioxidant defenses, as we've already seen, and signal transduction. All right, so now I'm going to do a little bit of research from my laboratory over the years, looking at how the thyrodoxin system and the glutathione system inside cells is regulated and responds to different stimuli. I'm going to tell you about some growth factor signaling, an extension of what Torn Finkel was doing. We're going to do spontaneous differentiation of an intestinal epithelial cell line, and finally a little of the work that we've done with NF-kappa B redox regulation. So the first story, this proliferation induced by a growth factor. So we know that there's this burst of ROS that's produced, and we know that that is a transient burst and it goes away. And so one question is, what is controlling the reversal of that signal? Is it a glutathione system or is it a thyroidoxin system? And so we developed a method to look at the redox state of thyroidoxin. It's a Western blot system where we can see the reduced form, the active site disulfide, and then under severe stress, we can see a second disulfide between two of those other cysteines. But that doesn't happen with EGF signaling. We simply see an appearance of more of the oxidized form early on, and that goes away, and it mimics the time course, the hydrogen peroxide burst very nicely. And we're going to convert this data into a number using the Nernst potential. And we're going to do that so we can compare it to other redox states. If you know a midpoint potential for any given couple, remember an oxidant and a reductant go together in a pair. That makes a redox couple. If we can plug those numbers into the equation, we can get a number that defines the redox state or the poise of that couple. And so when we did that, we saw that the cytoplasmic thyroidoxin 1 was the only one that became oxidized. And here the scale is in millivolts. The more negative you get moving up here, the more reduced it is. So these cytoplasmic thyroidoxin 1 is becoming oxidized, and then it's being converted back to the baseline reduced state. And we looked at some other couples. We looked at the thyroidoxin 1 within the nucleus, and that wasn't changing at all. We also looked at another thyroidoxin 2, which is localized in mitochondria, which I haven't told you about yet. I'm not going to talk much about. But it has its own reductase. It's a separate system. That one's not changing either. And the glutathione redox potential isn't changing. So what we have is a potential with this NADPH oxidase burst stimulated by the growth factor binding to its receptor. We've got the signal that could be oxidizing any of these four different pools, but it's only doing one of them. And so that leads us into different ways to think about the type of thing that's being oxidized and where it might be. The second model I want to tell you about is this model of differentiation that mimics what's going on in the intestinal microvillus, where stem cells are proliferating at the bottom, and they move up, and as they reach the villus, they differentiate into active epithelial cells until they slough off at the top and have to be replaced. And so that can be mimicked by plating these intestinal cells in a dish and just letting them grow. And we can see as the cells proliferate over time, this is a 28-day experiment, they reach a point where they're no longer dividing, and at that point, markers of their intestinal epithelial activity shoot up, and at the same time, the glutathione redox potential becomes more oxidized. So as they move from a proliferating state to a differentiated state, the redox potential changes. The thyroidoxin redox potential in this case hasn't changed at all. So this is the opposite of what we saw with EGF signaling, where only the glutathione redox potential has changed and not thyroidoxin. And the third story I want to tell you about is with NF-kappa B regulation. It's a transcription factor involved in inflammation. And it's normally sequestered out in the cytoplasm and associated with the inhibitor I-kappa B. And when it receives the right signal, the I-kappa B is phosphorylated and degraded, which allows the NF-kappa B to move in the nucleus and bind to the DNA. And through a great deal of effort, we were able to find that the thyroidoxin system actually affects the final step, that if you don't have a functioning thyroidoxin system, the NF-kappa B will move in the nucleus, it'll bind to the DNA, but it won't turn on gene expression. So to summarize, I've shown you 3 examples. I've shown you an example where the cytoplasmic thioredoxin pools selectively oxidize. I've shown you an example where the glutathione redox pools selectively oxidize, and an example where the nuclear redox is uniquely affecting a pathway. And so the take-home message here is that different redox couples regulate different cellular processes within different subcellular compartments. So we're already starting to see some of the complexity of the system and why there's not one pill that will fix everything. So now I want to switch and spend the rest of the time talking about what we've been doing with extracellular redox potential. This is a different set of problems. So inside the cell, we've been focused on glutathione and the thyrodoxin. And that's because glutathione is present at about 100 times greater concentration than this other redox couple, the cysteine-cysteine. Outside the cell is the opposite. There's 100 times more cysteine and cysteine than there is glutathione. So outside the cell, this is the predominant redox couple. And we can measure both of these, and we do, but we believe that this is the one that is having a more dramatic impact on cellular function. And to show you how cells and the extracellular environment can interact, I wanted to start by showing you a picture of some primary fibroblasts that we were growing in culture. And we did an experiment that other people have done with cancer cell lines, and that is we shifted the redox potential of the media. And we did that by putting in different ratios of cysteine and cysteine. And so down here at the more reducing end of the scale, we had a lot of cysteine. And here at the top end of the scale, we had mostly cysteine, the oxidized form. And 24 hours later, the cells have come back to the value of normal plasma. And you can see this even at earlier time points. It's a fairly rapid response. So the cells are actively involved in controlling their own extracellular redox potential. And like I said, that's been known. What was new in this study is we took fibroblasts from the lungs of a young mouse and fibroblasts from the lungs of an old mouse and did the experiment. And what we found was that the old mice had a new preferred redox potential. They also restored back, but to a different point. Here they went back to about minus 55 millivolts instead of minus 90 millivolts. So there's something different about the old cell that makes it either want to have or have this more oxidizing potential, or it's impaired in its ability to get back to the ideal potential. So one of the things we knew about the redox potential, because of some work that Alan Ramirez had done with Jesse, was that if you take fibroblasts and culture them in a reducing media or an oxidizing media, you can change the expression of this extracellular matrix protein fibronectin. And what we saw in our old cells that were more oxidized, we saw increased fibronectin expression, just like if you took a young cell and put it in an oxidizing media. So we're not sure which is affecting what, but clearly these two phenomena are closely tied to each other. And we wanted to see what might be responsible in these old cells for the differences. And we did a microarray study. We found lots of different genes. Turns out lots of extracellular matrix proteins are being changed in the old cells compared to the young. But when we looked at all the antioxidant enzymes and the thiol disulfide regulating enzymes, we found 36 of them in the list. Only 2 were changed between old and young, and that was the extracellular superoxide dismutase, we thought could be important, and the other was the cysteine-specific subunit of the XCT amino acid transporter. So it turns out that we're fairly certain that the SOD3 isn't contributing to this extracellular redox difference, but this almost certainly is. And so this is a transporter that brings cystine into the cell in exchange for glutamate. And the reason this is important is there doesn't appear to be a system outside the cell for converting cystine to cysteine. You can't do that reduction outside the cell. The cystine has to come into the cell where it's reduced to, we know two of the enzymes, they're both thyrodoxine reductase family members that can reduce the cystine, and then that can either be pumped out of the cell or it can be metabolized to glutathione, and the glutathione can get pumped out of the cell and broken down to cysteine. So we think that the extracellular redox state is being regulated inside the cell, but that the transport across the cell is a limiting factor. And so if we lose expression of this, then we lose the ability to funnel cystine through this intracellular mechanism. And when we looked, we confirmed by PCR that in fact the expression of the 7A11 is down. But more importantly, we could fix that. We used sulforaphane, a broccoli sprout extract, to upregulate the gene expression of 7A11 so we could bring it back up. And when we looked at the extracellular redox potential, we see that we also correct that. So we're pretty excited. We found an intervention that we can use, and this is being a dietary component, potentially could be translated into animal and clinical studies. And so to summarize what we're doing with the extracellular, I wanted to point out that we're now starting to move this stuff into thinking about what's going to intracellularly. And as a offshoot of that, our experiments are starting now to look at the interplay between the extracellular and the intracellular. And one of the ways that we're starting to approach that is with Mike Merchant and Jian Kai. We're doing what's called redox proteomics. You probably know about proteomics. You put all the proteins through the mass spec, see what proteins are different between two samples. Here we're looking specifically at the redox state of cysteines within those proteins. And we can do that by labeling the reduced and the oxidized with different tags. And we can actually mix together. We've got up to, I think there are 10 different tags you can use. Here we're using 4 of the tags, so we can mix all this together, inject it into the mass spec, and for every mass that comes off, the reporter ions are cleaved. Here's a couple of them, and you get a readout of oxidized in the young, reduced in the young, oxidized in the old, reduced in the old. And so you can get these ratios for every protein that you can identify. And I'm not going to get into the results that we got yet. This is still preliminary data. We've just gotten it. We're starting to analyze it. But we found over 200 proteins where we could measure all of those redox states for any given cysteine. And if we decide that a twofold difference is significant, so on this scale, I've shown the ratio of oxidized to reduced. So a positive number means that protein is more oxidized in the old cells than in the young, and a negative is more reduced in the old. And so of those 200 proteins, 100 of the proteins were actually oxidized, more oxidized in the old than they were in the young, and 9 of them were more reduced. And so we're excited about this, and we're actively looking into what these proteins may be doing, how they're going to be affecting the function of the cell. We're also doing a number of animal studies, so we can remove the plasma and different tissues from a mouse and measure their cystine and their glutathione redox potentials. We're doing a number of different animal models of liver and lung disease. And one of the things that I'm going to show you is that's come out of a number of these studies where we've included male and female animals, is that males are consistently more reduced than female mice. I don't know what to make of that yet, but the NIH has a real push now to consider sex and gender issues when you're designing your experiments. And I think that this is certainly going to be one of the things that we're going to have to work into our experimental designs moving forward. And I'm going to finish by talking about some of the clinical studies that we're doing. With Julio Ramirez and Paula Perrani, we're doing the plasma redox in patients with HIV and COPD, and we're looking at them at 6-month intervals of follow-up. We're doing another study with Kareem El-Kirsch looking at plasma redox as a potential biomarker of the effectiveness of oxygen therapy or CPAP therapy in obstructive sleep apnea. And then this isn't our data. This was published a few years ago by a Japanese group. But I thought it was very pertinent to some of the stuff that we're finding. They were looking at plasma cystine levels and glutamate levels in patients with cirrhosis, alcoholic cirrhosis. And the child score is increasing here, and the plasma cystine concentration is increasing. And at the same time, as the child score increases, glutamate is decreasing. If you remember, this key transporter we've identified is a cystine glutamate antiporter. And so this data would suggest that we've got a problem with the XCT cystine transport system in these patients. So let's come back to our original case study. We've got our 66-year-old male Hispanic ex-smoker with IPF. He's got a plasma redox potential that's about 20 millivolts more oxidized than a healthy control. So what can we do with that information? What does it mean? Does it mean that the fibroblasts in these patients are now more poised to make more fibronectin and other ECM proteins? Can we intervene by targeting this system? Is it going to be a biomarker of effectiveness? Is it going to actually mediate the effectiveness? If we switch that redox potential to more reducing, will we decrease pro-fibrotic marker production? And can we use this as a screen to predict people that will become more susceptible to disease in the future? And these are all questions that we don't have good answers for, but I think these are the right questions to be asking and to be thinking about. And I want to just close with this complicated-looking slide because I think it kind of summarizes some of the key elements and brings in one more level of refinement to the system. So we've been talking about all these different redox couples, from the most reducing NADPH, donating the electrons, all the way down here to our plasma cystine, which is one of the more oxidizing potentials. So within the cell, it's nice and reducing, and outside is more oxidized, but within the cell, there are compartments that are more reduced or more oxidized than others. And this is a scale showing a hypothetical protein that would be in equilibrium with one of these redox potentials. And if it has a midpoint potential of minus 210 millivolts, that means it's sitting here at minus 210, it's half oxidized and half reduced. If you move 30 millivolts in one direction, you get a 10-fold shift in the amount of thiol to disulfide. If you move 30 millivolts in the other, you get a 10-fold shift the other way. So relatively small changes in the redox potential can have a dramatic impact on the redox state of any protein in that environment. But this isn't the only protein there. There are proteins with midpoint potentials of minus 120. And so it's going to have a whole different set of situations where it's oxidized and reduced. And we have proteins up here. So we've actually got proteins that are sitting on this whole spectrum. And they're all going to be sensitive to this regulation. They're going to be turning on or off, or more susceptible to be turned on and off, depending on which compartment they're in and what's happening in that compartment. And so I just want to close by acknowledging, so I actually reached back to stuff I was doing in my postdoc. So this goes back a long way, and I'm sure I've left off a lot of the people that were involved in the process over the years. And I know already that I've left Paula off. She's involved in our HIV study, and sorry about that. But we've got a lot of people in my lab that have done the work over the years. Tom Bert and some students of mine, Yushuan most recently, has been doing a lot of the stuff with the XCT 7A11 study. The people in the Roman lab have been a huge help and have done a lot of this work. I've shown you some of Caleb's data on the sex of the mouse. And people from the Jones laboratory, when I was there, I worked with, I showed you some of Patrick's data with the EGF and Devon's data with the differentiation of the KCO2 cells. And then we've got a lot of other people that are involved. Some of them I didn't mention specifically, but the animal studies that we're doing with a lot of the people here. We're looking at the different animal models. I was able to show you some of the stuff that we've been doing with the Redox Proteomics, with the proteomics core here. And finally, I want to acknowledge the funding over the years from the NIAAA, the NIEHS, and most recently the General Medical through funding our COBRA award, of which I'm a part. So as Dr. Roman mentioned, the NIAAA has funded the University of Louisville Alcohol Research Center. He and I have a project on this kind of stuff together. And with the general medicine, we're looking at environmental exposures and diet, and particularly interested in how cysteine redox states are affected there. So with that, I'll close and ask if there's any questions.
Thank you, Bert. I really appreciate it. We have questions already in the audience. A couple of things. When I started doing a little bit of science, I promised myself that I would not get into research related to oxidative stress. To me, it was just too complex. And what we found is that you can't avoid it. It's unavoidable. It's everywhere. And one of the gratifying things for me has been to have Bert next door of my laboratory, because every time I come up with some stupid, simple answer to something, he brings me 27 of these slides, and I go back to my room disappointed and sad that I don't understand this well. And what I told him was that the audience was unlikely to be, including me, to follow every single aspect of what he described. But if you leave the stadium today, the auditorium today, with a little better understanding of how complex this is, then we would have been successful. And that's what he showed you today. So, Bert, one of the questions you have before we get into the audience questions is that you talked about oxygen radicals, you talked about final redox potentials, and you talked about this idea of letting go of the old concept of the ratio or the balance between oxidants and antioxidants, and learning more about disruption of redox sensing. How does a cell sense redox?
It's an excellent question. So particularly when we're thinking about like redox signaling, the thing that you think of immediately is phosphorylation cascades, where a kinase interacts with a specific substrate and phosphorylates it and changes its function. And we're looking at cysteine oxidation in an analogous term, except the specificity's got to come somewhere else. There aren't specific oxidases for each substrate. There's the oxidant production. So the specificity, we think, comes from the target itself. So some cysteines are more accessible out on the surface, and because of localized environment, will have a different pKa. And it's important because the thiol actually needs to be in the deprotonated or thiolate form to be reactive. So you can change the microenvironment to make a particular cysteine more reactive. But I think the most important thing you do is put the target near the source of the oxidant. So localization becomes a really important part of these signaling cascades, if you will, that involve oxidation. So the sensing, I think, really is you're sensing oxidant levels through differentially reactive cysteines.
Patients are more oxidized than the normal patients, number one. Second thing, we talked about how all these antioxidant treatments have failed. And then in these settings, can you give antioxidant treatment and then see the redox state change of the patient? Can you measure that so that has changed?
Well,
Those are two key questions. Can you repeat that?
yeah,
Every disease.
the question is plasma redox oxidized in every disease, and can we measure a change in plasma redox in response to therapy? And I think those are excellent questions that we need answer to. We know that in several diseases, or certainly in toxic exposures, that we can see a shift in plasma redox potential in people. People who smoke are more oxidized. People who take certain or who are on certain chemotherapeutic agents become more oxidized. But we are nowhere near in a position to say all diseases are associated. Just one, we need to look for those. And the second part of the question is, can we use it as some sort of marker of effectiveness of therapy? And I think that that's a real potential. No pun intended. But I think that's something that we should shoot for, especially when we're designing therapies that might particularly target this system. Then we would definitely want to be looking at this as a readout of the effectiveness of that therapy.
One thing that I found interesting is this concept that when we think about an oxidation radical, we think about a hit that happens in microseconds. Whereas when we think about oxidation or retitle, we're walking around oxidized for years. How do you test that in humans? If your experiment lasts 30 years with an oxidized systemic cytokine potential that may promote atherosclerosis or that may promote cardiac disease, I mean, this is going to be a problem. Let's go here.
So really good stimulation of this, and Dr. Arnold Spola was proud to suggest he had this type of remember the phase 5 cafeteria creative design. Anyway, specifically for the cardiovascular field, I looked at your graph of redox, plasma redox. How about the HIV patient versus the healthy control? What about exercise and also stats? Do they improve over the healthy controls? Has that been looked into? Because those are two things that
So the question was, has anyone looked at plasma redox in cardiovascular disease, particularly with exercise training or statin therapy? And as far as I know, no one's looked at that specifically. People have looked at oxidative stress markers, as I understand, but never this type of measurement.
we have proposed a role of the microbiota in the gut on redox rate in the intestine in the body. Do you know of any reason or evidence that the microbiota in the lungs is involved in
So the question is, like we know about the microbiota in the gut in redox regulation, is there a role for microbiota in the lung? And I think that's an excellent idea. I don't think anyone's looking into that, and maybe I'll take that one up.
So I have two questions about the cystine transport. You showed data from lung fibroblasts. So for the lung fibroblasts, the way to store cystine system reverse for the systemic perfusion. And the second question is that a singularly vital protein? Have you considered looking at SNPs to see if there are SNPs associated with pulmonary disease?
So that's two good questions. One is, are the lung fibroblasts driving systemic plasma redox uniquely? And secondly, have I looked at any SNPs in SLC7A11? And I don't know the answer to the first question. It seems unlikely that one cell type that we just happen to have looked at is the one cell type that's controlling global systemic. There's probably other cell types. One of the things that we're looking into is looking at endothelial cells, since they're actually in more close proximity to the plasma. And the second question in the SNPs, I haven't done anything looking into the SNPs and lung disease.
Dr. Roman mentioned 59 different studies that were negative different antioxidants. To my knowledge, nobody ever looked to see whether they were actually producing oxidative stress. So I think that's important. The question is,
So the question was, is it better to give an exogenous antioxidant or to induce your body's own natural antioxidant defenses? And I personally, given that we've tried a lot of give the exogenous and it doesn't work, I'm certainly of the camp that we should be thinking about inducing the endogenous defenses. And this example that I gave with sulforaphane, it's certainly inducing the 7A11 expression, but it's also inducing a lot of antioxidant enzymes. We have evidence that in terms of extracellular redox, that this particular enzyme being induced is the key regulator. But I think that the fact that we're also inducing peroxidases and glutathione transferases and reductases is certainly going to be a good thing. Yes. So the question is, why is, given that males and females are different, is there differences that arise during pregnancy or I assume other? So is it, I guess the underlying question, is it a hormonally regulated event? And we don't have any information on that. So those are good questions to think about there, absolutely. Yes. It is. That's all right. So the question is, why did sulforaphane restore 7A11 expression? And the answer, and she asked, is it an NRF2 target? And the answer is yes. That's why we chose sulforaphane to try to intervene there.
Any other questions? You had another one.
Follow up?
Go ahead.
We've only looked so far at expression levels of KEAP1 and NRF2. We haven't looked at localization or any other measure, and the expression levels are the same between old and young.
Any other comments? So I think the summary in my mind, Bert, is that over the past few decades, we've been focused on proteins up or down in disease. Then we focus on mRNA being up or down in disease. Then we focus on genes being up or down in disease. Then we focus on modifications of those genes up and down in disease. And you're moving us into the area with Mike and others whether the redox state of a particular protein is up or down in disease. And by looking at protein content, gene content, mRNA content of those particular proteins, you're not going to find a difference. It's really in the function of those molecules based on their redox state. And that's kind of interesting because that really blows all this up. And it really, before we try another multi-million-dollar trying whether NAC helps you or not, we probably need to sit down and think about this very carefully because of its complexity. Would you agree?
Yes.
Okay. Thank you, everyone. I enjoyed your Thursday. That was good. You did good. Yeah.
Oh, okay. Hello. Hi. So you're actually doing redox in the gut?
No, but we've shown that amyloid proteins made by bacteria in the gut enhance a whole series of inflammatory components which include INOS and NF-kappa B. And that could affect oxidative balance in the body. Right. But we haven't measured it. No. So I don't know if perhaps we can talk about how it can be measured in our next series of experiments.
And you're using mice?
We did this in rats. Aged rats.
Okay.
And by email,
yeah. Well,
I can send you our paper. And we're going
I would
to
have
be
to
doing it
borrow
again in transgenic mice with Alzheimer's and Parkinson's genes.
I can send you an email when I get back to my office. How about that? And then that way you'll have my email.
And so what is known about the role of the microbiota in the oxidative balance? Otherwise,
I know that people are doing it. I've seen people who actually look at the gut as a source of reactive oxidation. But I'm not really.
the main problem